Thank you for joining today's IAFP webinar, Practical Guidance for Validation Studies from Start to Finish. This webinar was organized by ILSI Europe, and it is sponsored by the IAFP Foundation. Today's moderator will be Erdogan Ceylan from Mérieux Nutrisciences. Before we begin, we have some housekeeping items. It is important to note that all opinions and statements are those of the individual making the presentation and not necessarily the opinion or view of IAFP. All attendees are muted. Questions should be submitted to the presenters during the presentation via the questions section at the right of the screen, and questions will be answered at the end of the presentation. This webinar is being recorded and will be available for access by IAFP members at www.foodprotection.org within 1 week. And at this time, I am going to turn the webinar over to our organizer, Angeliki Stavropoulou from ILSI.
Yeah, thanks, Dina. Thanks a lot. Oops, that is moving by itself. I'm sorry. Can you? Yeah, that's perfect. Thanks a lot.
There you go. I've given you keyboard and mouse control. Go ahead.
Oh, that's perfect. I'll just go to the beginning of the presentation. So yeah, welcome everybody from my side. Thanks a lot for being here today. I am Angeliki Stavropoulou, Scientific Project Manager at ILSI Europe, and I will shortly present to ILSI Europe through its Microbiological Food Safety Task Force, which organizes this webinar. So ILSI Europe is a non-governmental organization that delivers science-based solutions that improve public health and safeguard the environment. Our mission consists of 4 actions, and it all starts with collaboration. Collaboration between industry, academia, public sector, and beyond. And the MFS Task Force is a perfect example, and you can see also the expert group that work on the project that this webinar is based. Then we identify common challenges. This task force aims at investigating microbial issues in foods that are related to public health risks, and then facilitates the development of harmonized science-based approaches to predict and prevent those risks. Then we deliver science. We have some classical deliverables, which are peer-reviewed publication, and here is the one that the seminar is based on, is online, it's open access, you can find it. And of course, we have some more that are more focused on practical guidance for industry, such as our black and white reports. And we do not stop there. Our 4th action is communicate and disseminate the science that is produced. And this can be either at annual conferences, such as IFP, or online seminars or webinars such as this one. So with that, I will pass the floor to Erdogan, our moderator for today, and our speakers. Thanks a lot for your attention, and I hope that you all enjoy the webinar.
Thank you, Angeliki. Hello, everyone. Thanks for joining us today. My name is Erdogan Ceylan. I will serve as the moderator for today's webinar. I am Research Fellow at Mérieux Nutrisciences. Mérieux Nutrisciences is an international network of accredited testing and consulting laboratories in 27 countries with more than 100 locations. In my current role, I consult for the food industry on FSMA, HACCP, microbiological risk assessment, process control, and prevention strategies. As we get started, and throughout today's webinar, we will be collecting your questions to ask our experts. Please feel free to submit your questions by typing them into the chat panel. At the end, we will have a Q&A session. Today's webinar features 3 distinguished speakers who will provide insights into validation studies. Our first speaker is Dr. Annette Winkler. Dr. Winkler is Food Safety Advisor at Cargill, Germany. She has more than 20 years of industrial experience in various roles. She has implemented food safety standards globally across different categories of food. In her current role, she manages the food safety program of a wide spectrum of products and processes in Europe, Middle East, and Africa. She is one of the well-known process authorities in thermal processing of foods, microbiological risk assessment, HACCP implementation, process validation, and verification. She is also very active at ILSI Europe and IAFP Europe and US. And our second speaker is Dr. Roy Betts, Research Fellow for Campden BRI in the UK. Dr. Betts has over 30 years of work experience in the variety of food safety and quality issues. He is specialized in food hygiene, risk assessment, and troubleshooting solutions to problems. In 2019, he became the organization's microbiology ambassador. He sits on various committees and bodies such as the UK Food and Drink Federation, ILSI Europe, the UK Advisory Committee on Microbiological Food, Safety of Food, and he has been a long-term member of IAFP as well. And our third speaker is Dr. Heidy den Besten, Associate Professor at Wageningen University in the Netherlands. Dr. Besten obtained a double major in food technology and mathematics and completed her graduate work in food technology, specializing in food safety. Her research activities focused on pathogen ecology, interlinking functional genomics, and prediction of microbial behavior. She acts as editorial board member of Food Research International, International Journal of Food Microbiology, and Journal of Food Protection. She is member of the program committee of IAFP and also expert group member of the ILSI task force on microbiological food safety. She teaches at Wageningen University and coordinates an undergraduate food microbiology course in Wageningen and in Singapore. Now, without further ado, please join me welcoming our first speaker, Dr. Winkler. Unfortunately, she won't be able to make it today, but we have her recording, and please enjoy her presentation.
Hello, welcome everybody, and thank you for joining this webinar on validation, on process validation. Apologies that I can't really join in person this time, so you hear a recording from my side. However, my colleagues are there to answer all of your questions. So, and I'm starting here with the initial steps of how to be prepared for a validation study. Okay, validation. Let's start from the very beginning. What does it mean? The definition given by Codex Alimentarius starts with obtaining and evaluating scientific and technical evidence. That's the first phase. That a control measure, of control measures, or the food safety plan as a whole. So the control measures, the second phase, when properly implemented, is capable of effectively controlling the identified hazards. So that's important to keep in mind. It starts with the identified hazards, then you need to establish an effective control. Then you look at what is your control measure, you obtain and evaluate the evidence, and then you properly implement. So we go through step by step and start with the beginning, which is how do we identify hazards? The first thing to know is be specific. Don't give a group of bacteria. Give a species, be as specific as you can. Sometimes even strain helps to be really identifying a target pathogen and also working through all the different steps of the validation. In that respect, your hazard study is of utmost importance, because in your hazard analysis, that's exactly what you do. Also considering the intended use, you identify the target and the significant pathogens for your products. Then, having done that, you're also looking into epidemiological information, you look at surveys, you look at published literature on prevalence, on occurrence. This together leads you to your target pathogen. There's a lot of help out there in literature, and one of it is a table that's been published recently where you can different products, what has been found in terms of surveys, in terms of pathogens. Once with that information, also you can look at your different pathogens, and remember I said be specific. Don't say you look for Enterobacteriaceae, this is not a pathogen. but look for specific group like Salmonella, or Aztec. And when you look for Listeria monocytogenes, remember it's a different group of organisms. They may behave different, they have different habitats, and so they are of significance in different kind of foods as well. Now from there, how do we establish an effective control? So how many log reductions are sufficient to control the biological hazards? Again, look in the literature, can you find prevalence rates? Can you find quantitative levels at initial stage? So it's a lot of literature, you need to do a lot of searching, maybe looking at your historical data. Also a lot of epidemiological information, even reach out to experts, external experts as well. Look at what is known about an infective harmful dosage, about the consumption pattern. Consumption pattern of your food is important. It's a huge difference if people normally eat 10 gram, 100 gram, or a kilogram of it at one single dose. Then again, in order to help, commodities have been collected in a table with a target pathogen, and here even with the performance criteria that have been published either by regulation, either by scientific literature for the different food commodities, and that are applicable, even process parameters have been added here. This kind of information is also called safe harbors, where you have process criteria, process parameter that are established either by historical information, by legislation that has long been in place and proved to be successful in history, or by scientific information. Now, this is not available for all commodities, for all foods and products. And so in 2013, a workshop was done, especially low moisture foods. And as the outcome of the workshop was published, there was a consensus that for low moisture foods where there's no growth, like peanuts, a 5 log reduction would be sufficient and efficient to control the risk there. So that is also information you can use when you don't really have scientific data for your specific food and product. So watch out what's being published. Then with that, moving on to what can be the control measure, process considerations. When you look at your process, how you process your food, look at the steps that are most effective and most likely to control the hazards, because those are going to be your control measures. For those steps, then you need to understand the principle of action. So what is it that kills? What is it that has an effect on the microbes? Following this control measure, you also need to consider what is the potential of recontamination. Do I have an open process, open to the environment, where recontamination can easily happen? What precautions do I need to take that it does not happen? And also, clearly in your product, is there potential for growth? And if there is, how do I control this potentially? So let's have an example in cocoa production that relates to low moisture foods. Raw cocoa beans can come in with Salmonella as the most important and recognized pathogen of concern for cocoa. Then in the process, different ways of processing, but in the process you see different roasting steps before they are grinded, and then out of the liquor with pressing you get the cocoa butter and the cocoa powder very shortly. Now let's look where there's heat in the process. In the debacterization step is heat, in the roasting step is heat, fermentation step is heat, and roasting step here again is heat. So 4 heating steps you can potentially choose from. Now, which one do I choose? Besides heat, also moisture has a significant effect. The higher in moisture, the better the lethal effect on pathogens. Raw cocoa beans come in with max 8% moisture, so fairly dry. When I can increase the moisture, when I do increase the moisture in one of the process, then that might be the one I want to choose. So let's have a look. Debacterization uses steam, increasing moisture. Alkalization uses significant amount of steam and water, again increasing the moisture. So overall, from all those processes, I would choose potentially the debacterization or alkalization as my really step to control Salmonella coming in from raw cocoa beans. Both roasting steps are usually dry roasting steps. They are much less homogeneous than moist processes and also not so easy to control. So if I have a choice, I go for a wet heating process for different reasons. One being moisture has a better Lidl effect, and also dry heat processing steps are not so easy to control as are moist heating processes. Having talked about thermal interventions, it's also important for all the different heating processes we use that we know how the heat transfer takes place to make sure that the product is truly heated, or depending on where the contamination sits, is heated where the contamination is being hit and is really being controlled. So distinguish between the conduction, convection, this is traditional heating, but also the newer heating processes like microwave, radio frequency, and ohmic heating. You need to consider here that they work with charged molecules. So when you don't have charged molecules in your product, the effect might not be what you want. Then there are lots of non-thermal interventions. They are now used in industry for different purposes, starting from pulsed electric fields, going to filtrations, UV high pressure processing. All have different effects and all have limitations. Again, in order to help and guide people through this, there have been a table and an overview added with different of those non-thermal interventions, the considerations, what they can do, and their limitations, including the references. So that helps you a lot in terms of guidance to determine what you need to consider once you use one of those non-thermal interventions. So in essence, you really need to know your process very well. And you also need to know your process parameters that are important to deliver the effect on the microbes. Moist heat processes work often with moisture, time, and temperature. However, there can potential other factors also play a role, like the weight and like the pressure sometimes. For other processes, gas or irradiation can also be important, or even instrument specifics. I just don't know, because you need to find it out together with your engineers. So reach out to them, ask them how the process works, understand what are the parameters you need to control, and then from there you can proceed. From the process, moving on to the product, that plays an equally important role, because you need to understand the characteristics and their variability. Especially moisture and water activity, as mentioned before, also the product plays an important role. The higher the moisture, usually the better the kill effect. The composition of the product is as important, as well as the pH. Fat and sugar often have a heat preservative effect. And also for other interventions, the composition and the pH in the product are important. The same is true for how the product looks like. Is it a paste? Is it with a rough surface? Is it with a smooth surface? How dense is it? That all plays a role. So you need to choose what is the worst case when you go for your validation study. So you need to know what truly runs on your line, even once, not only for all what runs normally there for all the time. To illustrate that, there's a nice diagram here. You see the heat-resistant comparison of various bacterial pathogens. Salmonella is highlighted. The lower heat resistant in the liquid processes, the higher heat resistant in the more low moisture food area. And you see there is a huge difference between the product that are moist, that are liquid, like milk, versus chocolate and peanut butter. This is really a significant difference you would need to consider, you would need to know. But more details about that and the data usage will be presented by the third speaker in this webinar. Then with that, I come to summarize, specifically looking at the process. Remember, in the process, you need to define your control measure. What is the best place for the control measure? What is the best control measure, control step you have in place? And then you need to understand the principle of action, to also understand operational procedures and parameters and their limits you need in conjunction with the process capability and variability. And then you can perform a trend analysis to make sure there's no drift in one direction or the other that can get out of control. For the product, please consider the product constituents, the product compounds, fat, sugar, and salt specifically, antimicrobial compounds, water activity, moisture, sizes, the surface, is it rough, is it smooth? Also important, especially for continuous processes, is the incoming temperature. So what's the, especially when it comes to heating processes. So consider all those variables to be ready to go for a validation study once you have defined your target biology and your action. With that, I want to say thank you for your attention, and our next speaker will take you through how to do a validation study. Thank you again. Thanks for your attention.
Hi there. I hope everyone is now listening to me. I'll just pop my my camera on so you can see me. My name's Roy Betts. I work at Campden BRI in the UK, and I'm taking over from what Annette said and taking you through some of the aspects of running a process validation study. And just there we go. Just going back through some of the things that Annette said, you need to understand certain things before you ever think about starting a study. She mentioned a lot of these, but just to go back through some of the things you need to go through. You need to understand what you want to achieve from the validation study. And what do I mean by that? What do you want to achieve microbiologically? We're talking about microbiological achievement here. So you need to think about the target organisms you're aiming to eliminate, and that might be one or it might be many, but you need to know what the hazard is that you're trying to eliminate. From that, you need to understand what process objective is you want to achieve. And what I mean by a process objective is basically the log kill. How many reductions do you want to achieve of your target hazard? Now, you may be able to find that out from legislation, from guidance documents, or maybe even from your own risk assessment, knowing what the potential contamination levels are in the ingredients that you use. But you need to have that there because that's going to be key to understanding whether your validation has worked or not. What's the process objective? Then you need to understand your process parameters. Annette talked about this beforehand, but that's anything that's important to the process. So things like the pH of the product, the water activity, the levels of fat or oil, levels of protein, portion sizes, whether you've got particulates present within the product, and if you have, what the particle size is, volumes of liquids that you're going to have as sources or whatever inside the pack, and even the viscosity of those liquids. And not only knowing what they are, but also their critical limits. What's the biggest and the smallest particle size? What's the top and the bottom pH that you consider to be acceptable within the product that you produce? You also need to have a good working understanding of the process equipment that's being used, that you're going to run these products through, and the operating characteristics of that process equipment. Now, when we often talk about doing process validations, we often talk about worst case scenario or using a worst case scenario. And really, what is meant by worst case? Well, firstly, we always should validate under what we would call worst case conditions. Now, worst case condition is essentially any condition which will minimize the effect of the process on the microorganisms that you're trying to eliminate. As an example, it might be the lowest water activity, because we know water activity has a huge effect on heat resistance of microorganisms. The lower the water activity, the higher the heat resistance of a microorganism tends to be. with fat and oils, we know that fat will protect microorganisms from the effects of heat. So the highest fat level offers the greatest protection. The largest pack of particulate size, and when I say the largest pack, I don't mean that you're going to pack off 500 gram packs and 250 gram packs. I'm saying if you're packing off a 250 gram pack, that's not going to be exactly 250 grams every time. It's going to have a tolerance. What's the tolerance in that? It'll be the largest pack size because that's going to take the longest to heat up. So what's the largest pack size that you're likely to get? And the same with particulates, the largest particulate size, that's going to take the longest to heat up. The most viscous, it holds up heat transfer. So most viscous you're likely to get within the range that you have. The coolest points in the process and the shortest process time. So all of these things you need to understand within the process that you're going to validate. And that's what we mean when we say validate the worst case scenario. It's essentially saying this is the worst it can possibly be. This is when the microorganisms are going to survive the longest. So if we validate under worst case conditions, then we should always achieve what we wish to when we're running the process for real. So now we validate. Now we can validate in a number of ways, and we can often talk about in-plant validation, where we actually take the validation into the production plant using production equipment, and we run the packs through the real process and see what happens. Sometimes we might do laboratory or pilot scale studies, where we take the validation and we run it in a trial, in a laboratory, or in a pilot scale production area at a much smaller scale. And many people do a mix of this. They may start validation in lab scale and run it up into in-plant validation. But there are pros and cons to both of these types of validation. Shown in this slide, if we go down row by row, with laboratory pilot on the left-hand side and implant validation on the right, it's clear that if we use laboratory scale validation, we can use pathogens when we do that validation. No hazard there. We're in a lab, we can use pathogens and see what the effects of a lab-based process on the pathogens are. Clearly in the plant, we can't. We're not going to be taking pathogens into a reduction area. We have to use surrogate organisms. These are organisms which we will have developed a knowledge that their resistance to the heat, if we use a heat process, their resistance to heat is the equivalent to that of the pathogen. Moving on downwards, clearly in lab scale, death data that we get from the validation relates to the pathogens we've used. In the plant, the death data relates to surrogates. So we have to assume, we've done the previous work to know that that surrogate its death is very similar in kinetics to that we see with the pathogens. In the lab, we've got careful control of the processing parameters in the lab equipment. We can control it very carefully on lab scale. If we move into the plant, the process parameters are coming from the actual equipment we use in the manufacture, and therefore control might be a little less delicate, should we say, than we can achieve in the lab. But it's real. That's what we actually see when we run the process in the processing plant. Within the lab, we've got a degree of flexibility in the way we inoculate the organism into the food. The amount of food material we use is relatively small. It may be only hundreds of grams in total. If we move into the plant, we're inoculating our packed microorganisms only where we can within the process that we have. We've got limited places in which we can do inoculation into the product and into the process, depending on where we get access to it. And we're going to be using very, very large amounts of material because this is a full-scale process line. So we're going to be using hundreds of thousands of kilograms of material in order to do the validation we have. In the lab, we have flexibility in the lab use. We can more or less do the validation when we want. In the plant, the trial has got to be fitted into a production schedule. We've got to stop producing whatever we're producing on that line, clean it, do the validation study, stop, clean the process equipment again before we continue on normal processing. So it's much more difficult and costly and I say just generally difficult to fit that type of validation into production scale. When we get down to data handling in lab, we have to interpret the data for the in-plant comparability because it's been generated under lab conditions. So we have to interpret how it might fit into in-plant data type. In-plant validations, we don't need to interpret anything. We've used the production plant that's going to be used in the way it's going to be used. No data interpretation really needed. We've got what we will get using that particular plant for production. So things you've got to do when you do validation. First of all, any equipment you use, whether it's lab scale or in the process plant, has got to be serviced, calibrated, and working correctly. We've got to have correctly working equipment. The inoculum that we use, we're going to use an inoculum of the hazard, or we're going to use a surrogate. That's got to be matched or adapted to product characteristics. We've got to be quite careful. If we take an inoculum that's been grown up in a microbiological broth at a nice pH of 7 under good conditions, and we inoculate into a product that's got a low water activity and maybe a low pH, some of those organisms will die through shock. Now, that's not what you want because that's going to skew or bias the validation and give you incorrect results. So you've got to make sure your inoculum is matched to the product. And that usually means suspending the inoculum in conditions that are as similar as possible to the product you're going to inoculate into. So adjusting pH, adjusting water activity. When you do the inoculation, you've got to inoculate the organisms into a realistic position. So if it's likely normally that the product is going to be surface contaminated, the inoculum will go on the surface. If it's likely to be internally contaminated, the inoculum's got to go inside. You've got to think very carefully about inoculum volume because you don't want to change the product characteristics. So you don't want to take 100 grams of product and inoculate it with 20 mils of the inoculum. Changes the product characteristics and the validation is not right. So you've got to inoculate very, very low volumes of inoculum so you don't change the product characteristics. And then you've got to think about the inoculum level that you need. That's going to depend on your process objective. What's the log reductions that you're going to get? Generally, we'd think about having your inoculum 2 logs higher than the kill that's required. So if you're looking at a 4-log kill, you'd inoculate with 6 logs of your target organism or your surrogate. You can't think about inoculating with 3 logs and looking for a 5-log kill because you'll never see it. You've got to inoculate 2 logs higher than the kill required. Then you've got to think about where am I going to inoculate? Am I going to inoculate in the production area or in a laboratory close by? And then you've got to think about, once I have inoculated, how do I transport my inoculated product to the processing facility? You've got to think about the maintenance of the viability of the inoculum. What you don't want to do is inoculate, and because of changes or conditions during transport, your inoculum is going to die or indeed grow. You want to maintain the level of inoculum that you achieved when you inoculated. So the idea about how you get your product, the inoculated product, to the production area is key. How do you transport it, maintaining the level that you've inoculated? Then how much do I need to do? How much is involved in validation? You've obviously got to replicate. You can't just do this once and get 1 data point. You've got to replicate. The more variability you think you have in your process, the more replication is needed to make sure that your validation is valid. Replication, what we tend to mean by replication is independent batches of product and independent batches of inoculum produced, not simply the same batch done at different time points. It's got to be independent batches that you go through with replication. Gives you the variety you need to make sure the validation is okay. If you're doing lethality studies, where you're looking at different time points, so you've applied a process and you're looking at what happens at different time points, you've got to think about a minimum, absolute minimum, 3 samples at every time point as a level of replication. If you're doing endpoint studies where you're running something through the whole process and just measuring what happens at the end, then it's more like 5 to 10 samples that you'll need to be sure of getting valid results for the validation. You've got to ensure you've got enough replication to give confidence in the results you get. And remember, you always need controls. You've got to have controls there to prove that what you're doing is correct. As an example, when you've inoculated your product and you take it into the process area, always take a control away and test for the level of inoculum at that point, just to make sure that you know that when you're actually applying the inoculated material to the process, the level of inoculum is what you think it is. Make sure you've got controls in place. Always remember, you don't do validations very often. The safety of your product depends on it, so make sure it's good. When you think about placing the samples in the processing line, if we're doing an in-plant validation, you've got to think about where and how you introduce them. It's got to be realistic. You may have limited access to points on the line, but make sure you introduce them in realistic positions. If for any reason you're using sample holders when doing a validation, and often when we're doing validation of dry materials, we may actually place the inoculated dry material in some form of sample holder so we can see it when it comes out the other end and we know that it's had the right process. But we've got to make sure if we use sample holders of any sort that they don't affect the process. So you've got to do some previous work on that to make sure the sample holders themselves, if you use them, don't affect the process in any way. At the end, you've got to collect the material that you've processed. And when you've collected it, you've got to make sure you then get it to a laboratory that's going to do the microbiology work to give you the data you need on what process, what level of reduction you've had. And you've got to make sure that at the end of that process, when you capture and transport the material, what you do minimizes changes to microbial numbers. You've got to transport those process samples to a laboratory quickly, and you've got to make sure the laboratory work is going to be done quickly. Because there's only two things microorganisms are going to do within a food material. They're going to die or they're going to grow. And you don't want either of those to happen in your material. You want the level of organisms to be exactly the same when measured as what was there at the end of the process. Once you've done that, you've got to think about the process environment. If you've done in-plant processing, you've got to clean up the process environment because you're going to be using that to process product that's going to go out to consumers. So it's got to be fully cleaned. You've got to audit that the specialist equipment that you took in to do the validation has been brought out. What you don't want is any of that special equipment being left around and potentially being a hazard in your normal production. So audit the equipment you take in for the validation and audit that you brought it out again. You've got to make sure the validation has no adverse effect on any subsequent normal production you do in the production plant. When it gets to the laboratory and you're starting to test the processed product, make sure the lab is using recognized methods and nothing experimental. Make sure that if they're available, recovery methods are used. Remember, these organisms have seen a process and some of them may be injured. And injured organisms may die and not be visible to you when you do your microbiology. So recovery methods that allow the recovery of injured microorganisms are key. Things like the use of selective microbiological growth media. That produces an adverse pressure on injured microorganisms, and potentially injured organisms may not recover. But it's important that we actually are enabled to detect the presence of those injured organisms, because probably in the real product, they would recover and they would produce a hazard to a consumer if consumed. You've got to decide whether you're only going to test for the organisms that you inoculated in, or you've got to look for background flora as well. It's a decision you need to make. And then you collect all of the data that you have in preparation for data analysis. Last thing, what about revalidation? You've done it once. Does that mean it's done? How long does validation last for? Does validation validity run out? I think what you've got to think of is that for validation, for any validation to remain valid, all of the equipment that you use in the process has to remain constant. It has to be constantly calibrated, it has to be constantly serviced. It's got to be operating in exactly the same way as it was when you did the validation. Any change to equipment, product flow, product characteristics, methods of operation, any of those things has to be reviewed to assess if revalidation is required. And even if you believe that all of that remains the same, it will be very prudent to revalidate the process at regular intervals, just to make sure that you know that process is working the way it was when you did the initial validation. Well, that's the end of my talk on how you do a validation. I'd like to turn over now to the next part, which is how we handle data, to Heidi, and she'll take you through some of the data handling options.
Thanks, Roy. I will take over. And my name is Heidi Besten from Wageningen University. I'm associate professor here in Wageningen. And I'm going to talk about how you can obtain scientific evidence as part of a validation study. And when you have performed a validation study, how you can evaluate the data and to evaluate if you have reached your target reduction. So as Roy mentioned, a very important element of validation study is collecting observational data. And also already addressed, you have to choose a worst case scenario. And you have two ways of testing, the lab scale and the in-process testing. And the big pro of lab scale testing is that you can use pathogens. However, if you cannot mimic your process in a lab scale environment, you have to proceed with in-process validation testing. And then you have to use surrogates, and then a part of your validation study will be that you have to check that your surrogate is exactly behaving as your target pathogen. Another way of collecting data or evidence as part of your validation study is collecting scientific data from the literature, which you can use to support the design of your validation study. And one of the challenging things is when you look for data in literature, how representative that data is for your product of interest. And also you will see, if you're going to dive into literature, you see a lot of variability in the data you can collect from literature. And one strong point from data collection is that you can also point or pinpoint to most important variability factors. And I'm going to explain that also today a bit more in detail. When you collect data, of course, you can use it to build a database. And you can use your database to perform a meta-analysis. And a meta-analysis helps you to predict the efficacy of your control measure. And I will also explain an example how you can apply a meta-analysis and predict then the output also of your control measure. So what are the different sources of scientific data? First of all, of course, the historical data of your own company. If you have performed validation study in the past, you can use this for reval if you want to revalidate or redesign a study. Second is that you, of course, you can use data already available in the literature. So scientific data published in freely available literature sources. Also, microbial risk assessment as published, for example, by the WHO or governmental agency, they are often a very, very rich source of kinetic data. And you can use that kinetic data for your own purpose. Also, a database I'm really fan of is Combase, which is like a freely available database with a lot of kinetic data. For example, inactivation data of different pathogens in different products. So you can extract these data. It's really open accessible, and which you can use as a first indication how effective your, for example, heat treatment will be. So a meta-analysis is a way to systematically compile and analyze a large collection of available data, in-house data, published studies, or databases, with the aim to produce a global estimate of a parameter of interest. For example, your d-value, so the decimal reduction value. But because you're collecting a lot of data, you also collect information about the variability of your kinetic parameter of interest. So a meta-analysis helps you to point to important factors that influence your kinetic parameter, but because you have an idea on the variability, you can also quantify variability. So the DZ concept is widely used to describe the kinetics. And here you see at the left side how you can estimate a d-value. So at the left side, the figure shows the log counts as function of time, and then the inverse of the slope is the d-value. So the d-value is the time you need to get a 1 log reduction. When you plot the log d-value, so the 10 log of the d-value as function of temperature, the linear correlation allows you to calculate the z-value, which is again the inverse of the slope. And the z-value actually is the temperature increase you need to reduce the d-value with a factor 10. So if you have a d-value at a specific temperature, for example, 60 degrees Celsius, and you also have the z-value, which can be, for example, 5 degrees Celsius, you can predict, having this d-value at your reference temperature and the z-value, you can predict the d-value at any other temperatures of your interest. For example, at a higher temperature at 72 degrees Celsius. So the d and z value allow you to predict inactivation kinetics at your temperature of interest. But when you look into literature, you find linear inactivation kinetics, but also non-linear inactivation kinetics. Curvatures can have like a concave downwards curvature or a concave upwards curvatures. And if you find data like this, and if you want to use it to, for example, estimate a d-value, you first have to look critical how these data have been obtained. Is this like curvature not like an artifact of the way the method has been applied? You have to rule out that like the shoulder curvature, for example, is caused by cell clumping, or that the shoulder curvature is caused by short heating up time, and that the temperature at the time you start measuring is not exactly yet at your set temperature. The tail, of course, if you see a tail in your inactivation data, then you should be sure that the tail is not caused because the counts are below the detection limit. So if you extract data from literature, make sure that you check if the method has been correctly applied. But there are ways to use this data. If their kinetic data are like with a curvature, you can use the Weibull model to fit your data and to estimate a d-value, because the Weibull model is actually an extension of the linear model with an extra shape parameter, which is the beta parameter. And by fitting this model to the data, it allows you to fit both curvatures, the concave upwards and the concave downwards. But as Roy also mentioned in the previous presentation, often you want to have, for example, a 5 log reduction with your treatment, or a 6 log reduction. And this advocates to not model the decimal reduction, the first decimal reduction time, but, for example, the 6th decimal reduction time. And you can do this by fitting this parameterized model to the data. And when you fit this model, highlighted here on the bottom, when you fit it to the data, the output you get is a time for a 6 log reduction. So I mentioned that you can use literature to obtain a first global estimate of your kinetic parameters of interest. And we have done a big meta-analysis study in our laboratory here in Wageningen. And what you see here on the slide is d-values of Listeria monocytogenes as function of temperature. And you see we got many d-values from literature, 940 d-values, and they came from different products. So d-values from Listeria in different types of products, and also cultured different ways. And you see that there are a lot of different variability in the d-values extracted from literature. But what you also see is that there is also a difference between products. And what the strong point is of a meta-analysis, that it can point to very important factors. And as Roy also mentioned, the water activity of a product has a huge impact on the d-value. And you can see here in the figure, the gray symbols highlights the d-values of Listeria in low water activity products. And you see that the d-value of in these products really deviate from the d-value in other types of products. And by doing this meta-analysis, you now, next to knowing that the water activity has a clear input, you can also quantify it. You know now what is the order of magnitude your d-value will increase when the water activity of your product will decrease. And these lines represent a 95 prediction interval of the data, and this is a measure actually of the variability encountered of the data in the literature. And the set value obtained from data as such is something you can use for making the predictions of the d-value at other temperatures. If you have enough data on a specific product, you can proceed on a more detailed level. And in this case, we had a lot of data d-values of Listeria in milk. And here you see the d-values as function of temperature for only in milk. And then, of course, you can have a much more precise estimate of the d-value at your reference temperature, and also the estimation of the set value, which both you can use to predict at your own target temperature. However, it's good to be careful when you extrapolate. So the set values are when you are calculated based on the data you have. However, when you want to estimate a d-value outside the temperature range of which your data have, then slight deviations in your set value can have a large impact on the prediction or the estimation of the d-value at higher temperatures. So an alarm bell should ring if you want to estimate a d-value at a higher temperature than where you have data on. So it's always good to check if the available data is in the range or out of the range of your actual situation. And if it's out of the range, use it with caution, because you can have then a significant under or overestimation of your d-value. So next, that products, the intrinsic parameters of a product can have a clear impact on the heat resistance of an organism. Also, strains which belong to the same species can vary really different. And that's also what we have been quantifying here in Wageningen. We quantify the impact of strain variability for Listeria monocytogenes as example species. We collected 20 different strains from all different sources, isolates from the industry, but also lab strains, so a wide selection of different strains, as diverse as possible. And all of the inactivation treatments we did always in technical duplicates. So running the experiment with the same culture, but using technical duplicates. And what you see here when you do performing technical duplicates, the data look rather comparable. However, there's also another source of variability, and that's when you reproduce an experiment really independently. And this is what you see here in this data. All the L experiments we have performing for the 20 strain, we always used 3 biological independent reproductions. And you see that the variability introduced by biological variability is higher than experimental variability. However, when we tested all the 20 strains, you see that there's a big difference between the strains. The most robust one is much robuster than the most sensitive one. So when you go to select a strain for your validation study, again, remember it should be in worst case scenario. So then it's good to select a robust strain of the species of your interest. So the pathogen of your interest, select a strain which is robust. And what is then, how big is the impact of strain variability? Now you can use a meta-analysis to benchmark the impact of this strain variability. So again, this is the figure I have shown you before, all the D values of Listeria from different products, and also different strain. And here you see the 20 strain we have quantified at exactly the same method, exactly the same way of treatment. And you see that this impact of strain variability contributes for a large part to the observed variability, indicating that strain variability is an important element to take into account. And again, therefore, you have to select a robust strain for your challenge test. So when you have used scientific data to design your validation study, and when you have performed your validation study, then you get your data out of it. So assume your target is to have a 5-log reduction. And this is the data you get out. You have performed 3 replicates, and each of the 3 replicates you have 3 samples, and each of them you have an initial count, so an initial inoculum count, and you have a final count. So a way of analyzing the data is doing it in a deterministic way. Then you take the average of a replicate at t is 0, and you take the average of the final counts, and then you get the reduction achieved. And then when you average the 3 replicates, you could come to the conclusion, okay, I meet my 5-log reduction target. However, in this case, you didn't address the variability in the data. So a better way of looking at the data is doing the minimal reduction case approach. Then you take the minimum log count at N0. So in this case, for example, for the first replicate, it's 7.9, and a maximum number of the final counts, which is in this case 3.25. And then you calculate the reduction for each of the 3 replicates, and then you will have to conclude that you don't meet your target reduction. And then you have to add an extra safety margin, or heat longer, or increase the temperature. So to conclude my presentation, and also some take-home message from the 2 previous speakers, available data is very useful to support the design of your validation study. But available data in literature doesn't always perfectly match the actual situation, and you have to decide with your validation team, your whole team, if you have to proceed with a challenge test, and often that's the case. And challenge test is required to prove the sufficient reduction obtained. And then recall the messages also given by Roy, that you have to address the variability with respect to samples and also batches, and select a robust strain. So having said it, I want to thank you for your attention, for being present, and then I give the floor back to the convener, Erdogan.
Thank you, Heidi. Thank you, Roy. Thank you, Annette. We have a good number of questions from attendees, and I will pick several of them. We have a few minutes left. And one of the question is the use of minimum reduction values versus average reduction values when we run process validation studies. Are we using minimum log reduction values as the worst case scenario versus the average log reduction when we do data analysis?
Maybe I can take that question.
Yeah, question is that when we run a validation study, are we using a minimum log reduction values versus average log reduction values as the worst case scenario?
Yes, as mentioned in the example how to analyze data and how to use data, you should always assume the worst case scenario. So the minimum reduction you achieve is something you have to judge, and if it doesn't meet your target reduction, you have to add an extra safety margin. So you have to higher the temperature or increase your time to make sure that you reach the aim of the target.
Just to add to that, hypothetically speaking, if we don't have any variability in culture or inoculation or inoculated samples, the average log reduction and minimum log reduction should be identical. But we are dealing with biological materials, so that's not always possible. And typically a minimum log reduction is recommended. I hope this answers the question. There's another question, Roy, this is addressed to you. What are your feelings regarding the potential presence of viable but not culturable organisms after a process and control step?
I see you speaking, Roy, but I don't hear you. So maybe I can take the question. So one way of making sure that you get also the the damaged cells is by plating on selective media with a cover layer of non-selective media, or the other way around. So by having that, you can ensure that damaged cells have the ability to also form a colony on your plate, and therefore also that you are able to count it. Because if you will only use selective media, damaged cells might not grow out to a colony, and then you miss it, and then you have an underestimation. So a combination of selective and non-selective media is then recommended.
Sorry, I lost the ability to turn mute off. I'm back now. Just repeat the question again. I sort of missed it in that in my absence period. It was to do with damaged cells and recovery.
Basically, the question was about culturable versus non-culturable cells when we run process validation studies.
Yeah, I sort of as Heidi said, really. You've got to really do the work before you start doing the validation, but by understanding which is your target microorganism, which might be the pathogen, it might be a surrogate, you've then got to work out with a microbiologist what the best way of ensuring you've got recovery of injured organisms. And in some cases, you would be able to plate out, if your product is fairly sterile to start with, you may be able to plate out directly onto a non-selective medium. In many cases, you'll have to go out onto some form of selective medium. So minimizing that selectivity is the way you have to go. And Heidi mentioned ways of doing that with overlays and so on. But the best thing to do is talk to a microbiologist beforehand and work out the best ways of ensuring recovery of injured organisms in your particular case.
Thank you, Roy. We have multiple questions in terms of culture selection and strain variability. We are running out of time, but this is actually a very tall question to answer. Mm-hmm. Do you have any insights in terms of strain selection and variabilities among strains?
Well, it again, typically microbiology answer, it depends. Yeah. I mean, often when doing validations of any sort, you may not inoculate with a single strain. You may inoculate with multiple strains as a cocktail to try and, I guess, span particular issues you might get with single strain variability or single strain effects. So you can look at using multiple strains of the same organism in a cocktail to try and ease the effects that you get of strain to strain variation. But besides that, it's really going with the expertise of knowing what the resistance of particular strains are to whatever control measures you have within your food. Because we might not be thinking just about heat, but you may have a low pH food or a low water activity food. So it's trying to pick the strains that most, I suppose, would be most able to resist the controls that you have present within your food product.
And maybe as an add-on, there also I think literature can really help, because you can use it as benchmark of the strains you want to use to see, okay, is it really a robust one, or is it based on the available literature, is it an insensitive one? That's also how available data can support selection of your strain.
And just one last note maybe to add on, when we talk about strain variability or the worst case scenario strains when we use for validation studies, we need to choose the most heat resistant strain for that product and process, but it doesn't mean that most heat resistant strain for other products. If we are dealing with meat products, most resistant strain could be different than that products with low moisture. Let's say if you are doing peanut roasting, most heat resistant strain of salmonella will be different for peanut versus meat products. So that should be considered as well.
Yeah.
I think we are out of time. I want to thank our presenters, IAFP, ILSI, and all attendees. And we will answer. We have multiple questions needs to be answered. We will answer those questions later individually, and the presentation will be available within a week. There were a couple of questions regarding if attendees will access this presentation later. The answer is yes. Presentation will be available, I believe, in 1 week. And that concludes our presentation. Thank you, everyone.
All right, that concludes today's webinar. We would like to thank all our panelists today. This webinar is being recorded and will be available for access by IAFP members within 1 week. If you're not a member, we encourage you to go to our website to learn more about becoming a member, and all IAFP webinars are supported by the IAFP Foundation with no charge to participants. Please consider making a donation to the IAFP Foundation so we can continue to provide quality information to food safety professionals. Thank you everyone for joining, and have a great rest of your day.
Thank you.